showcasing some of our tools, specifically our tools for slicing and dicing the data, including clinical and medical imaging way of creating cohorts. And then we'll also show our genomics tools. So hopefully it will be an interesting meeting for you. And we're definitely looking forward to showing you what we have. With that, I'll turn it over to Alina. Alina, please go ahead.
Thank you, Alex. Good morning, everybody. While I'm sharing my slides, I'm going to briefly introduce myself. As Alex mentioned, I am a physician by primary training, but here for the program for TB Portals, I serve as a portfolio manager, helping Alex run day-to-day operations of our program, as well as engage with the new partners and ensure that we are growing and maturing daily. All right, so it is my pleasure to introduce TB Portals. We are an open access data repository. What that really means, that we are a large program that includes, of course, tuberculosis cases, as well as a suite of tools, in addition to all the scientific support we provide. For the scope of the program, and just the way we outlined it at the beginning, we designed it to focus on pulmonary TB, specifically multi-drug resistant tuberculosis, is our focus of interest. However, as the program grows, we're expanding it to other types of tuberculosis. I should also mention that we initially started focusing on adult cases, and now we are engaging in more with the pediatric world, although it is challenging in one ways, but opportunistic in the other ways. So we are a global conglomerate and international network of scientists and clinicians, and our ultimate goal is to, of course, advance TB research, improve global health through knowledge, through collection of very good cases that can really unveil and give us a peek and insight in this century-old disease. We are celebrating our 10 years anniversary, so we are a middle schooler. Lots of attitude, lots of knowledge, still lots to grow, lots to understand and learn. We are spanning across 24,000 cases. I need to update my slides. In 20 countries, across 7 continents. We have 60 medical center sites that are providing their data to us, including Eastern Europe, Asia, Latin America, and Sub-Saharan Africa. And before I advance, I wanted to mention this tool that I'm demoing here on the right. This is tuberculosis in 3D, and I will come back to that, and I would like to demo that separately. That's one of the real-time tools that we develop to showcase and demonstrate progression of tuberculosis and its treatment in a 3D environment, where Medical Illustrator rendered this actual close to nature, less of the science fiction, but actual true rendering of the disease pathology throughout the case. So we've been noticed, specifically TB Portals over the years have been part of several strategic plans, including the U.S. National Action Plan for Combating Tuberculosis, NIH Strategic Plan for Data Science, and I will cover that in a minute with the new NIH Data Science, science-focused data sharing policy. But we all mentioned several times at the United States National Action Plan for Combating MDR, TB, and NIAID strategic plans, WHO strategic and TB research innovation initiative. We've been included in many, we've been named as several scientific leaders, to name the few. So what makes a good database really good, in my opinion, again, as a clinician, but also I'm biased towards this program, but overall, if you just think about what is the good case, what makes it valuable, I think it's the complete data inputs. So in our program, we set a bar for folks to provide 3 different data types that are pillars for each case that we have: genomic, radiomic, and clinical data. What it means that for each case we collect, we require at least 1 X-ray or CT scan or whatever the standard of care in the country, as well as pathogen genome. We sequence the bug, we look closely at the lineages, we look closely at the origins, and I think my colleague Michael Harris here, who is with us on the line, will cover that in the detail. And of course, the clinical component, the clinical case itself, including the socioeconomic, the laboratory, treatment progression, all sorts of different results that could be pertinent. We store our cases, of course, in a de-identified manner. We make them publicly available over time, not right away, but over time, for researchers and clinicians across the world to conduct their research, their analysis through a simple process of data use agreements. I wanted to really pause and talk about the composition of each case and what the database represents. So as I mentioned, we are nearly 24,000 cases right now, including almost 3,000 of CT scans. And of course, we know, and I want to put it up front and center, that CAT scans are not a standard of care in outside of the United States and Canada. They're very expensive. We are working in Ukraine, Moldova, Belarus, and India, and Mali, for example, to name a few countries. And so CAT scans would be economically prohibitive. So folks are usually getting a chest X-ray, and we have about 20,000 chest X-rays. And you see here that a subset of them has been annotated. What annotation really means is where a radiologist completes our standardized descriptor for each and every case, and then it's used for us to analyze it, as well as for our radiomics team to utilize AI and other pertinent newly developed cutting-edge technologies to bring a power of machine learning and computing into analysis of these cases, again, in our push to really be able to advance science in that way as well. We are engaging with industry partners such as QAI and others to develop those annotations. We have about 7,000 of the whole genome sequencing for the pathogens. We have additional subset in the pipeline. There's always some sort of a delay with the getting the genomic sequencings back. We are very picky about the quality of our genomes. So when possible, we sequence them locally in the country, but in the countries where they don't have a good infrastructure or correct setup in place to sequence the genomes, we actually partner with a large hub of a huge lab in Borstal, Dr. Newman's lab, to service as our sequencing hub, and we sequence them centrally in Germany. So that, of course, requires all the logistics, getting the samples from countries there, getting them the information back, and we, of course, have infrastructure in place. And from our clinical cases, we have a good number of biochemistry results. And I'm not talking about cute things like, red blood cells and glucose input. I'm talking about a true biochemistry markers that can be assistant in analyzing the case. For example, your ILTs and ASTs, your liver markers, your kidney failure markers, especially in the multi-drug resistant regimen with the harsh therapeutics. We wanted to assess patient compliance, and we wanted to see was, for example, comorbidities or pathology or situations related, adverse events related to the therapies that were like contributing to a person's ability to sustain the drug regimen and how that reflected. So we collect all sorts of things. We look at liver enzymes, whatnot. And we have about 140,000 individual test inputs of those in our database. Our program, I believe, is matured into not just being a database, but also come with a suite of tools. We think our data is complex and easy enough to be engaged with and analyzed. However, we wanted to really provide a support and umbrella of services to our users. Therefore, we developed several tools, and I know that some of them you will have a pleasure of seeing today, including Depot, that's our multi-domain cohort building tool, our GAP genomic application, our AP radiomic application, as well as the TB in VR and 3D that is great for overall patient education, medical students education. Well, let's just say even some clinicians enjoy seeing those actual renderings, as well as, of course, our main capturing portal, which we call Case Browser, where scientists can provide their results either in an aggregate way, and then we just upload them on the back end, as well as we have several screens where folks can just manually put their data in. We all about data sharing and access. We think that you can have the best data in the world. If nobody knows it's there and nobody uses it, then you might as well not have it at all. So our goal is to make TB data openly available for the research community worldwide. And in a few slides, you will see just how many people across the world use and enjoy our data. We have tools that can visualize the data, conduct analysis. We also have a number of experts. I think 40 folks on the team have a PhD or some sort of a credential to themselves to be able to help and analyze the data. And of course, that comes at no cost to our users. We are a government-funded entity. We just want the research to be better. All right, so talking about the program, and I mentioned we are about 10 years old. So how does it all started? I like a good story. So it started in Belarus, where there is a staggering number of resistant tuberculosis. And it grew exponentially from the Eastern Europe. Of course, we know after the fall of Soviet Union, we got the data that tuberculosis is a major problem there. I grew up in Ukraine myself, know firsthand how bad things are in the country. So we started growing it from Eastern Europe, but then we really looked at the map and we realized there's pockets of TB elsewhere. So we expanded, of course, to African continent, as well as Central Asia, growing farther through Eastern Europe region, and lately in Latin America. And I think here on the map, there's few countries that we're working right now that we're trying to engage is in Latin America and African region that could be great contributors of data. So I mentioned that your data is only as good as the people who uses it. So we started tracking our data use agreements, and we've been steadily growing the number of institutes and the number of companies out there, private and public, as well as industry and academia who uses us. And so here on the map, you see each dot represents at least one medical center, private or public company that request our data to be used. And this is the map of our data users with the obvious hubs in the United States as well as in India. And in the United States, we made analysis and we saw a very good distribution between a solid, your conservative academic centers, as well as the industry partners and, Google and other places, to name the few, who really see value in our data. That translated nicely, I think, in the publications and citations, and you see our stats right here. Publications both internally done by our scientific team, as well as externally, of course, done by our data users and partners. We've been well cited, and I think our track record speaks for itself. When I briefly mentioned at the beginning of my presentation about the data management and a sharing policy, I was very intentional about that. We are well aware of that now for each and every grantee out there who are going to entertain NIH grants. They are required to deposit their data in a publicly available, shareable environment, and we think TB portals can be just that. It's not just a data dump where you put your data, to check the box and meet the requirement and just be done with it. We feel there could be a really good opportunity for folks to enjoy the infrastructure that we build around the data, as well as be able to really engage with their own data and compare it to the data in the TB Portals program in the database itself, as well as find some meaningful results. This is our webpage, and of course, I'll drop it in the chat. So if time allows, I'd like to maybe spend a few minutes and demonstrate one of the tools that we have, the TB in 3D tool, as well as how to engage with the database itself. So here, one second, sorry, there you go. I just wanted to double check, are you able to see my landing page of a screen?
Yes.
Excellent, thank you so much. So here this is our landing page of our website. Please come see us, visit us. And here we have the TB in 3D, and I pulled up one of the cases here, but I just wanted to demonstrate this is not an isolated case. We have a library of several cases here, 12 cases, I believe, to demonstrate to you an actual patient histories. And some of them even have pictures associated with surgeries. But here, for the sake of today's, I wanted to present to you a case of actual TB case of this patient and the tool itself. So the tool is interactive. You can, of course, click through and remove the structures. You can keep only only lung tissue. You can remove bronchial tree if you'd like. You can play around with removing and including the infiltrates and all the lesions back and forth. We have 2 annotations, manual done by our medical illustrator, as well as AI enhanced. And you can see the AI is doing a good job, but it's pretty rough around the edges where the manual annotations are so precisely done by somebody who is a beautiful artist, but also has a training in biology. And here, for the sake of this presentation, I wanted to share with you this case of a 46-year-old male person, formerly incarcerated, who who came in initially with this presentation, and then he came back later, and then it was really bad, and so he was 5 months into the treatment like this, and then he was lost to follow-up, and then it just really expanded. So you can see the dramatic proliferation of the TB, and and I think this case did not end well for the patient. So I would like to share with you the TB portal's data capturing portal itself. So here I'm just taking to a real case. We can see who the person is, the gender, the region, country of origin, as well as some of the stats, education level, risk factors, all the usual suspects for TB, as well as what kind of specimen we had from the person, the sputum, the microscopy and culture input, as well as the drug sustainability testing. Here we can see resistant or sensitive to which medication. As well as here's the example of those biochemistries that I mentioned. We can see the person was in treatment, how long, and by what medication. Had lots of images, of course, because of the severity of the case. I mentioned we do the pathogen genomics. Here's the genomes, the lineages, and you can see them in the sequence archives at the National Library of Medicine. We did a TB profiler here. We have a timeline of the case, how it progressed, and what happened. And then I think he deceased at the end. We lost him to this severe disease. But this is a case at a glance, and this is all the data points you are able to obtain. So with that, I think I'm okay to pause. Would it be better? I'm just trying to understand, would it be better to answer any questions right now? Or should we carry on with our demos and then do the Q&A at the end? Rajita, what do you think?
Why don't we keep going? If people have questions, I'll ask that they put them actually in the chat, but then yes, it would be great at the end if we can stop for some verbal questions as well. So let's just keep going and we'll look for some questions in the chat as well.
Excellent.
Thanks.
Sounds like a plan. All right, thank you so much. And with that, I would like to hand over to my colleagues who will be presenting the depot and the genomic tools. Drew or Michael?
I'd be happy to go. Hi, everyone. My name is Drew Hoppus. I lead our data management within the TB Portals program, so all the collection, data quality, and data sharing within the program. We have a group of data engineers and software engineers in Depot that work year-round to maintain all of our applications. Today, I'll be walking through a couple of our tools, as Alina said, to complement our great database. We have a few different software tools that perform different functions and specialize in different analytic use cases. And so I will start. Give me a second, sharing my screen. Beautiful. You see my screen okay?
Not currently seeing. It just says you started sharing. Oh, here you
There
go.
you go.
Yes. Yeah, now it's coming through.
Okay, great. I'm going to start with
It's hard to see the elements on the screen. Do you mind zooming in on this?
Sure thing. Sorry, gang. I think my Zoom crashed out there. Should be back up and running. Can you see my
Yeah, it looks nice.
Yes.
Oh,
Great.
wonderful. Thank you for your patience. So I'll start by demonstrating our radiomics analysis portal. This is a tool that allows users to define and save groups of chest x-rays based on specific descriptors. Then you can explore and share individual x-rays, browse patient case data, and review images over time. We really wanted to create the remote radiomics analysis portal to really have a sandbox where users can test things in a domain-specific way for radiologists and image researchers. And so this tool is really aimed towards imaging specialists and students, and it draws really from well-defined cases, including radiologist image descriptions. So we collect labels that really power this particular tool. We'll start the first particular function within the radiomics analysis portal that we'll look at is find conforming images. And so we can select here different sextants of the lung. And this is 6 different regions, and this is the level at which our radiologists are annotating our images. And so by clicking here and selecting, I'm going to select large cavities here. You can see that it updates the upper right sextant. And if I were to click here and do the same thing there, what it will do is it will return all images as a searchable query back to me that have large cavities within our database in the upper regions of the lung. And so it will return this nice table with an ordered list. Whenever the weight is 100, all the requirements of the query have been satisfied. And then from there, we can select cases, build cohorts, and save those cohorts for analysis. One thing to note with this is a sextants are one of the unique things about our TB Portals database, and it really is, this whole tool is driven by those manual clinical annotations that we require for every one of our x-rays. We also have 3 different services, AI services from Cure AI, the UIIP Institute in Belarus, their imaging lab, as well as this organization called Zine that produces complementary AI predictions for abnormalities on images. So this is really a search function to help you query the database and build popular cohort sets that you'd like to analyze. This brings me to the next function within the tool called Find Similar. And so it does exactly what it suggests, which it uses a similarity algorithm under the hood to, when you take a local external image that's separate from our database and upload it to this tool, it will provide you back a list of results from our database that the similarity algorithm suggests is the most similar to the image that you have. So I'm going to take a local image. This is a patient with tuberculosis. I'm going to ask it to give me back 25 results. And as you can see, it was a very easy drag and drop. And what it will do is return that list of 25 images along with some attributes, some descriptive attributes that let you know a little bit about those cases. And so from there, you can do a number of different things. You can create a cohort. You can go to the case browser tool and investigate more detailed case notes about a patient. But what I will do in this case is I'm going to click View Graph. And it brings up this network visualization, this graph visualization, with my outside image at the center. And then you can color the different nodes of the 25 returned similar images by different categorical data that allows you to learn about what those similar images, what the distribution of key information about that cohort that's returned is. So in this case, we have, we're coloring the metadata by outcome. Good news here is that out of the 25 returned results, it looks like the majority, were cured cases, or cases in which they completed treatment with only one failure out of the 25 results. So just one example, you can color by a number of different things if you'd like, but another great way to use our database as a researcher, if you're really, in this case, perhaps somebody that's interested in walking through similar cases and seeing those case notes, that might inform a case that you're interested in locally. This would be a good tool for that. Here, bring up the service called Predict TB. So this is a classification service using neural networks to predict TB. For historical reference, this was originally built for Brazilian partners that wanted to automatically triage prisoners, and they wanted to do fast screening. So used a large training set for this. And while this service is not unique, other companies like Cure.ai have services like this that predict TB. This service is free. And so I will take that same image that I just used for similarity and upload it. And in this case, it gave a strong confidence score that indeed it does predict that it has TB. If I were to use an example on, oops, this is the wrong format. Give me a moment. If I were to take, I took an image that this is actually a chest X-ray with pneumonia, and uploaded it here, and it's pretty strongly confident that it is not TB. And so it's something quick that could be used. The last service I'm going to make note of, and then I'll transition to our depot tool here would be our time series explorer. And so I will take a case here. Actually, I need to log in for this one. For the sake of time, I'm going to skip past this, but just want to let you know you can take a patient case that perhaps you landed on through find conforming or find similar X-rays, and through that patient ID, you can bring up timelines which show different treatment regimens and where in the timeline of a case are they taking medical imaging. So this can give you a little bit of a sense of the context of those particular images and what course in the treatment they were in. So I'm going to transition from RAP here. Like I was mentioning, RAP is domain specific. It is really centered around the images. The tool I'm displaying here, this is what we call our depot. This is the data analysis platform for the program. And so its primary functions are to easily build cohorts through an easy-to-use query builder and the use of clinical genomic and imaging similarity algorithms. After you've built cohorts, it's really easy within this tool to study those cohorts and perform hypothesis testing. Today, I'll be providing 2 use cases to give you a feel for how to use the tool. One is going to be studying the discrepancies between genotypic and phenotypic resistance classification. And then we'll look at some of the differences between the WHO bedaquiline treatment regimens, and specifically, we'll be comparing bedaquiline resistant and bedaquiline sensitive patients and seeing whether there are any statistically significant differences between those populations. The first cohort use case, though, is looking at that genotypic and phenotypic sensitive resistance classification. So in order to do this, let me zoom in a little bit just to make it a little easier to see. Okay. You can see on the left-hand side, I'm just pointing and clicking, and here you'll see clicking on the type of resistance, and I'll click sensitive patients here because I want to compare the phenotypic and genotypic resistance classifications. I'm also going to click and genomic data exists. And so what that will do, you'll see that this querying syntax has automatically come up into the query builder. And all I have to do is execute the query, and it will slice the data down to the 1,339 patients. These are just the patients that are publicly available that fit the parameters of my query. And so now that I've sliced it down to that group of patients that are clinically defined as resistant, usually doctors will perform a DST test at baseline to support that classification. Now that I have that information, we can compare it to the genotypic sequenced data to see if there are any differences that we see where we might have resistant cases that are shown by the drug susceptibility test that actually are showing genotypic resistance. And so what we'll see here is a bar chart and a heat map where this particular bar represents one SNP, and these are SNPs with known associations with tuberculosis, resistance to tuberculosis drugs. And so in this case, this is the SNP RPSL, PELAS 43ARG, and almost 5% of our cases that, we are labeled as having phenotypic drug resistance are showing this SNP, which is associated with streptomycin resistance. And so from there, it's aligned with the heat map, where by hovering over these particular cases, we can see individual cases that and what their result was, what their SNP was. And so by doing this, and I'll skip ahead, one of these SNPs, we actually take a look that we got from this heat map was for patient 739. And so patient 739 actually had an outcome of failure. And so if we wanted to, we could go to, this is the patient case browser, which allows you to look at, as Alina said, the information for a particular patient, like you're looking at case notes broken down by each individual data domain that we collect. And if we look in this case, we can see that for the DST testing, they performed a BACTEC test. It had results for isoniazid, rifampicin, and ethambutol, where it was sensitive for each of those. And while the genomic sequence came up and said that it had streptomycin resistance, or a SNP associated with streptomycin resistance. In this case, we look and we see that the treatment started at day 8, and they actually lucked out in this case because they did prescribe rifampicin, isoniazid, and ethambutol as the main drugs within this regimen. So I share that use case as an example of all of these tools perform different functions with our database. And so, you might be able to start with Depot and you want to drill down into an individual case to gain an insight and gain context. It's very easy to use information from these different tools. It's all based on the same data. And so once I have my cohorts built, you can also save those cohorts very easily by just clicking the Save button. And then once you do so, you can access those particular cases in a save page. And so in this case, I've already made for our use case number 2 cohorts, one with patients with bedaquiline resistance, demonstrated by the BACTEC test, and one with bedaquiline sensitivity. One thing of note, you can see that there's a very small number of patients so far that are demonstrating bedaquiline resistance, only 66 out of only over 3,600 results, but they do exist, and we certainly can study them. And so can quickly go to our cohort comparison tool. And this would be how you can see in cohort B, it already brings up my betaquine resistant patients. And I can access my saved cohorts and compare those as well. So for the sake of time, assume that I've clicked run on this. It will produce results that that can show the difference.
I apologize, it's
Yeah.
8:52, and we also have Michael Harris, just for you to be aware of time.
Absolutely, yeah.
Thank
I'll
you.
wrap this up in 1 minute. You can just see that here we have BACTEC resistance to some of the other drugs that are in that 2022 WHO regimen here with clofazimine, linezolid, and so moxifloxacin. So you can see some associated resistance. And then also, interestingly, we're seeing statistically significant difference in outcomes. And so between that and all of these different tabs, you can get different statistics from. One that I will just show very shortly is that in this particular view, the full genome view, you can see the SNPs that are most statistically significantly different between the two populations. And so, if we wanted to start looking at which SNPs are showing are associated with resistance between these two populations, you can do so and utilize the data and perhaps extract the data. So it's a powerful tool. It's very easy to use. If you're at all interested in this or would like to get further training or help, please reach out. I'd be happy to do that. But I will pass the ball to Michael here for the last demo. Thank you.
Thanks, Drew. Lena, I just wanted to check with you. I know you want to have some time for question and answer as well. So
I think let's do our best for demoing, because I'm really excited for the folks to see the tool, and then we will tackle the Q&A. But we don't want to cramp your style.
sounds good. Okay. I am going to show. Can you all see my screen okay? Is that coming through okay?
Yes, we can see it.
Okay, great. So I'm going to talk about the TB Portals genomics team and our efforts. So I lead the genomics team. My background is in computer science and math. We have a number of scientists, life scientists on the team as well. So we have a diversity of experience working with the tuberculosis genomic data. And just to summarize, these are some things that Alina and Drew mentioned, but we have over 6,000 genomes deposited in the NCBI Short Read Archive, SRA. We have different kinds of samples. So most of our samples are paired in Illumina reads, but we have done some long read sequencing as well in PacBio, and have done full genome assemblies with that PacBio data. We have worked with mixed infection samples and actually have some tools for identifying mixed infections in genomic data. We've worked somewhat with the duplex sequences and targeted sequencing, and we're also getting into sequencing of non-tuberculosis mycobacterial infections. So just quickly, I'll go through this very quickly. Let me see if I see I'm not in presenter mode. Oops. Let's see. I think how's that? A little bit better, hopefully. So our sample processing workflow is that we get the DNA, which is extracted in country. That goes usually to a sequencing center. We do a lot of our sequencing at Borstal. And once the DNA is sequenced, we get FASTQ files delivered to us, usually through FTP site or other means. We do quality assurance checks on those FASTQ files to make sure that the samples are not contaminated, and other types of tests, and then we submit those to SRA. Once the variants have been called, once we've gotten those FASTQ files, we map the reads to the reference genome, the TB reference genome, and this is our variant calling pipeline, which I won't go through in detail right now, but the output of our variant calling pipeline is really the input to the tool that I'm going to talk about today, which is the genomic analysis portal. And in contrast to the tool like Depot and the radiomics portal, the genomic analysis portal is meant to really dive deeply into the genomic data and allow for on-the-fly analysis of sequencing data and integration with other TB portals data types, but it's really focused on the genomic data. And so this is a screenshot from the first screen that you'll come to. If you go to the genomic analysis portal, you can look at different aspects of variants, resistance prediction, and then there's an integrated genome browser. So for resistance prediction, we rely on external tools for doing those predictions. We're using TB Profiler, which is a tool from Jody Phelan in the UK, who developed this tool and is a very prolific software developer. It's used by many people for doing resistance predictions. There's also the microbe predictor, which is the Iqbal Lab produced that. So you can run various resistance prediction tools on the data and get reports about the resistance. So on this screen, this is an example of a sample that's been run through several different tools. And we can see that the concordance about the resistance predictions based on different variants that are found. This is a comparison report on the right. And then on the left is a detailed resistance report that's coming from TB Profiler. So this will show which drugs the sample is predicted to be resistant to, the variant found, and then some other variants that have been found in the genome as well in resistance associated genes. I see that I'm at the 9 o'clock hour. Are we okay to go a little bit longer?
Yeah, if you want to take a couple of minutes, that should be fine.
Yeah, okay. So another tool that we can execute through this is a distance network of between samples. So we can have groups of samples that you can actually look for relationships between the samples in terms of genomic distance. And so the next few slides are examples of networks that were generated by the genomic analysis portal. And these can be colored by metadata. So for example, the lineage or country of origin or outcome can be used to color the nodes of the network. This is another example. And you'll notice that in the parameters for this, so we can specify the sample distance threshold to look for, so there will only be an edge drawn between the nodes. Each node is a sample if the distance between those samples is, in this case, 15 or fewer SNPs. We can get in more detail so you could see actually this tightly coupled network where if you click on the edges or mouse over the nodes, you'll get more information about the samples and the relationships. I'm going to skip forward a little bit, and we can also, do very detailed analysis about variants contained within a cohort of samples. We have integrated genome browser where you can actually bring the data, your own data, into the NCBI genome browser, and can do also a GWAS analysis similar to what Drew showed, but you get it's a little bit more detailed in terms of the genomic information that is provided and kind of allows with integration with the genome browser to look at various positions in the genome that are of significant differences between cohorts. We've also done work in our long read sequencing efforts to look at assemblies of different samples that we have, looking for large structural rearrangements and other things that can be found with long read sequencing, looking in detail at regions in the genome that are difficult to sequence with short read data. That has been really, PacBio data has been really good at looking at these large structural differences between the samples. And then I'll just highlight a few of our analyses that the genomics team has done with different collaborating countries. So this is a paper which we published looking at a cohort of samples from southern India. We published that in collaboration with Dr. Mahadev Rao. This is a analysis of reactivation versus reinfection on some samples that we collaborated with on from Moldova with Dr. Valeru Crudu, and some of our long read sequencing data, which we use to look at short read accuracy of mappings of short reads. And we did this in collaboration with Max Marin and Maha Farhat at in Harvard. So I think I'm about out of time, but I want to leave a little bit of time for questions. And thank you very much for your attention.
Thank you so much. I think there's been a number of questions, and Alex, thank you so much for answering those as we go along. That's been really, really helpful. So I think if there's additional questions, perhaps we can try and have them sent to you all just to save some time. I know we're losing some people because of we've hit the 9 o'clock hour. But please feel free to reach out to the folks at the TB portals. And the other thing we were thinking about with TB Report is to actually have a small working group so that we can actually get these collaborations going. I think that would be really great if we can try to sort of have a little more interactivity so we can actually make things move a little bit forward. So I think, Alina, you shared the information for TB portals as well as the information for all of your contacts. I know, Eric, you saved that, you share that as well. So we would really appreciate if you could all contact them. And if anybody wants to join with Rick to join some like a steering committee or anything like that, where we could actually get this moving forward, please feel free to email the main Rick email address, and that should be super helpful. I want to thank you all again, Portals team. This was absolutely fantastic. We really loved the hands-on demonstration. I think it really helped. And there's just so many things that we can be doing with this data from a teaching and research standpoint. So we really, really appreciate your time and presenting such a mammoth project in a really short time frame. So again, thank you again. We did record this, so we will try to share this with others if you need to be able to see that. Thank you again, everyone. Take care and have a great rest of the day, evening, wherever you are.
Thank you for the opportunity to speak with everyone today. And like you said, we have our contact information if there's any questions, comments, or anyone's interested in collaboration opportunities.
Thank you for having
Fantastic.
us.
Thank you very much.
Thank you,
Thank
Aline.
you.
Thanks, everyone.
Thank you. Bye-bye.